Hello, my name is Liisa, and my talk is about chemical variability of apatite in the shells of fossil lingulate brachiopods. It is known that skeletal apatite has a very variable nature, and because of that, fossil skeletons can yield different kind of information, for example, biochemical, environmental, and diagenetic information. The question is, where in the skeleton are the areas with the information we need, and how this information could be preserved. In case of fossil brachiopods, it has been shown by means of powder X-ray diffraction studies that there can be two different apatite phases in one fossil brachiopod shell. One of these phases is similar to the apatite in recent lingulate brachiopods. The other phase is different. The existence of two different apatite phases in one fossil brachiopod shell means that the apatite chemistry must be changing inside of fossil brachiopod shells. And in my study, I wanted to know how the apatite chemistry is changing in fossil brachiopod shells, where we can find these two apatite phases and how different these apatite phases are. To study this, I used the shells of Ungula ingrica, which occur massively in North Estonia. I collected them from the Iru outcrop. They can be found from the Kalle formation, which has been assigned partly to Furongian and partly to Vahur division. For comparison, I used the shell of recent Lingula anatina. I molded the shells into plastics, and I studied the polished median cross-sections of the shells. Under scanning electron microscope, the shells of Ungula ingrica look usually massive, with only the hints to its original shell structure, which should be composed of alternating compact and porous lamina. Here is the ideal formula of hydroxyapatite. However, skeletal apatite is never with ideal composition. Every part of this formula is open to substitutions. For example, calcium can be substituted by other cations like sodium or magnesium. Part of the phosphate is usually substituted by carbonate anions, and hydroxyl anions are most commonly substituted by fluoride anions. And to study this kind of variability, I used a combination of two methods. One of the methods I used was infrared spectroscopy, which allows to study the existence and nature of phosphate, carbonate, and hydroxyl anions. And the other method I used was energy dispersive spectroscopy, which allows to study the distribution of single elements like sodium, magnesium, calcium, or fluorine. And I used mapping regimes with both of these methods. Here are the results of infrared mapping. And here we can see that the apatite chemistry is changing inside of fossil brachiopod shells, and in this case, these changes are following the brachiopod shell lamination. These infrared maps show that there is more carbonate anions in the apatite in porous lamina, and less carbonate anions in the apatite in compact lamina. Here is another example of infrared mapping, and these infrared maps show that there is more phosphate anions in the apatite in compact lamina and somewhat less phosphate anions in the apatite in porous lamina, where there was more carbonate. Carbonated apatites are actually very common, and both biological and sedimentary apatites are usually carbonated apatites. So to study the nature of carbonate anions in apatite crystal structure, it is useful to take a look to the variations in infrared spectra. This is how infrared spectra differ in recent Lingula natina and in the compact and porous lamina of fossil Ungula ingrica. In general, all these spectra look quite similar. However, here are also some differences. And I would point out the differences in this carbonate region. In this region, the infrared peaks for recent Lingula and for the compact lamina of fossil Ungula are at similar locations, both at about the wave number 873, which means that at least part of the original carbonate environments may have been preserved in the compact lamina of fossil Ungula. However, in the porous lamina, this carbonate species has a completely different location at the wave number 864, and the exact nature of this 864 carbonate is not well understood, but this carbonate appears to be frequent in fossil apatitic skeletons. And it has been suggested that the appearance of this carbonate species in fossil apatitic skeletons means that the apatite in fossils have been diagenetically changed. On infrared maps, these diagenetic and original carbonate species have very strict locations. The diagenetic carbonate anions are strictly in porous lamina, and the original carbonate anions are strictly in compact lamina, and maybe even more intensively in the central parts of the compact lamina. Elemental mapping under scanning electron microscope also showed that the apatite chemistry is changing, and these changes are following the Brachiopod shell lamination. For example, in compact lamina, we have less fluorine and calcium, but more sodium and magnesium. And these elements are actually the ones that substitute for calcium in the apatite crystal lattice, so these elements are like compensating the lack of calcium in the apatite in compact lamina. This is how compact and porous lamina look like under scanning electron microscope at high magnifications. One of the criteria for the good preservation of fossil skeletons is that the apatite crystal size must be small. And on this photo, the apatite crystal size in compact lamina is comparable to the apatite crystal size in recent lingulate brachiopods. In porous lamina, however, the crystals are much bigger. So we have several chemical as well as structural aspects which suggest that the apatite in compact lamina and the apatite in porous lamina have a different origin. The apatite in compact lamina is represented by original skeletal apatite, which has been recrystallized but may still have preserved some of its original characteristics. The apatite in porous lamina is mostly represented by orthogenic apatite, or the mixture of skeletal and orthogenic apatite, which may have been further altered by diagenetic fluids because of the porous nature of these porous lamina. The first burial of the shell probably was quick. Some of the original shell fibrillar organics may have been preserved, as it has been described, for example, in case of Cambrian Ungula inornata. And these phosphatized fibrils could have acted as additional nucleation sites for orthogenic apatite, and when these orthogenic apatite has filled the porous lamina, then it could have protected the compact lamina from further recrystallization. So to come back to the questions of the presentation, which were how the apatite chemistry is changing in fossil brachiopod shells, where we can find these two apatite phases identified by means of powder X-ray diffraction studies, and how different these phases are, I could conclude that these phases are quite different. I listed these differences in these two columns. The apatite phase 1 can be found mostly from compact lamina, and it can be interpreted as skeletal apatite, which could have preserved some of its original characteristics. The other apatite phase can be found mostly from porous lamina, and it can be interpreted as orthogenic or secondary apatite. And if we need some original information from fossil brachiopods, then the most likely place to find this information would be compact lamina. So thank you.